Welcome, everyone. We're delighted to have you join us for today's webinar from Dyne Therapeutics, who'll provide an update on the Forzetto phase 3 study with Z-rostudirsen, Dyne's exon 51 skipping drug. I'm Michael Kelly. I'm the Chief Scientific Officer at CureDuchenne, and I'm going to get us started with a bit of housekeeping. Firstly, you have an opportunity to ask questions today, and you can click on the Q&A button at the bottom of the screen and submit a question anytime throughout the presentation. There'll be a moderated Q&A session at the end of the webinar, where I'll be joined by my colleague, Dr. Liana Orlando, CureDuchenne's Vice President of Research. And for those questions that we may not be able to address, we'll work with the Dyne team to follow up with you. And after today, this webinar will be available on CureDuchenne's website as a resource for the community. So let me begin. For a refresher on the company's FORCE platform that's used to target muscle and deliver the exon skipping oligonucleotide, I'll refer you to the Dyne webinar CureDuchenne hosted last December with Dyne's Chief Medical Officer, Dr. Doug Kerr. That video can be accessed on the CureDuchenne website under the Community tab. You'll remember that Dyne reported that the registrational expansion cohort of the phase 1/2 DELIVER clinical trial met its primary endpoint. The company reported a statistical, significant, and robust increase in muscle-adjusted dystrophin expression of 5.46% at 6 months in patients dosed at 20 milligrams per kilogram once every 4 weeks. This was followed with an impressive muscle-adjusted 18.3% dystrophin expression at 12 months, and together was accompanied with functional improvements across multiple clinical measures out to 24 months. That data served as the basis for filing a biological licensing application, a BLA, in May for potential US accelerated approval. The BLA is currently under priority review at the FDA, with the PDUFA date set for January 21. That's approximately 5 months from now. And if approved, it will be the company's first commercial product. The Forzetto study is planned as a confirmatory trial to support US and ex-US marketing applications. And to talk about this phase 3 study, we've Dr. Maria Naylor, the Vice President of Clinical Development at Dyne. So without further ado, let me hand it over to Maria to get us started.
Great. Thanks, Mike. And good morning and good afternoon. My name is Maria Naylor, and I'm the Clinical Development Lead for the Duchenne program at Dyne Therapeutics. I'm also joined today by a couple of colleagues, including Julia Carpenter-Conlon, who is our Patient Advocacy Lead at Dyne, as well as Jordan Messer, who is our Duchenne Global Medical Affairs Lead. And I'd like to start off by thanking CureDuchenne for giving us the opportunity to present this webinar, and to all of you who are dialing in either live online or listening to this recording for joining us today. So I'm thrilled to be able to speak to you about our Forzetto study, which is the phase 3 trial of zolbetuximab in people with Duchenne muscular dystrophy with genetic variants that are amenable to exon 51 skipping. My goal today is to provide you with an overview of the Forzetto study and then to answer any questions you might have. So before I go into the details of Forzetto, I'm first going to provide a little bit of background information on zolbetuximab or Z-rostudirsen, which is a little bit shorter and a little bit less of a mouthful, which leverages the FORCE platform for broad delivery of an exon 51 skipping PMO to tissues impacted by Duchenne. So we know that Duchenne affects a number of different parts of the body, skeletal muscle, including the diaphragm, smooth muscle, cardiac muscle, as well as the CNS. And one of the goals of a Duchenne disease-modifying therapy should be to prevent and potentially allow for the repair of the damage done to these tissues. One of the ways of effectively doing this is to bring that therapy directly to those impacted tissues. And Dyne as a company was founded on this principle of targeted delivery. So how do we do it? Well, one of the ways that we've done this is that our amazing scientists have developed an antibody fragment called a Fab that binds to a protein receptor called TFR1 that is expressed on the surface of different tissues within the body. They also developed a special nucleic acid molecule called a PMO that's designed to enable the production of near full-length functional dystrophin. And so this PMO payload is attached via a linker to the Fab fragment, and it's these three pieces together that make up z-rostudirsen. And we have now both preclinical as well as clinical data that show us that z-rostudirsen is doing what it was designed to do, which is when it's infused in the body, directly binds TFR1 and brings that PMO payload directly to those tissues impacted by Duchenne. So we first studied z-rostudirsen in the clinic in our phase 1/2 DELIVER study, which initiated back in 2022, with the goal of identifying the registrational dose of z-rostudirsen and to assess its safety and efficacy in both ambulatory and non-ambulatory participants. DELIVER also allowed us to collect the data for a BLA submission for U.S. accelerated approval. The current status of DELIVER is that it remains ongoing with participants in a long-term extension, which allows us to collect data to better understand the long-term safety and efficacy of z-rostudirsen. We also have a PDUFA date just 5 months from now of January 21st, 2027. So the data from the DELIVER study were used to inform the design of our phase 3 Forzetto study, which we're going to talk about today. The goal of Forzetto is for it to serve as a confirmatory trial to support the potential conversion of accelerated approval to a traditional full approval in the United States, as well as to support global access and reimbursement. And the current status of Forzetto is that we have sites initiating globally with one active site currently in the US. So in the next slide, I'll take you through the key design elements of Forzetto. And I'll focus your attention on the slide to the top half here. So Forzetto was designed to enroll approximately 90 ambulatory males aged 4 to 18 years with Duchenne amenable to exon 51 skipping. Participants are randomized 1-to-1 into a 72-week double-blind placebo-controlled period in which participants receive either z-rostudirsen 20 mg per kg every 4 weeks or placebo. Upon completing the placebo-controlled period, participants will have the opportunity to enter into an open-label long-term extension in which all participants will receive z-rostudirsen 20 mg per kg every 4 weeks for up to 96 weeks. So the bottom half of the slide details some of our key eligibility criteria. And in the bottom left-hand box here are a list of our select inclusion criteria. So first, participants need to be ambulatory upon entering the study, which we define as being able to walk at least 8 feet or 2.4 meters without the use of orthotics and without assistance. And so what this translates to is a score of a 1 or a 2 on the North Star walk item, which is the second item of the NSAA. So that means if a score of 1 or 2 on the second item, by our definition, is an ambulatory participant. And if a participant happens to score a 0, then they would be considered non-ambulatory and therefore not eligible for the study. Other inclusion criteria include an age of 4 to 18 years at the time of informed consent, a rise from floor time of less than 10 seconds at both screening assessments, and all participants need to be on a stable dose of steroids for at least 24 weeks prior to randomization. The box on the lower right-hand side here details some of our key exclusion criteria, which mostly focus on potential concomitant medications that a participant might be on and the associated washout periods. So first, any alternative exon skipping or dystrophin modifying therapy would need to be washed out within 24 weeks prior to randomization. Eteplirsen only requires a 1-week washout. And givinostat or any other ongoing immunosuppressive therapy other than steroids requires a 12-week washout period. Additionally, any pharmacologic treatment other than steroids that might have an effect on muscle strength or function also requires a 12-week washout period. Gene therapy is not allowed at any time, either before or during the study. And we do allow for the prophylaxis or treatment of congestive heart failure, but we do require that any medication that's administered for this is stable. Either the medication itself and the dose regimen are stable 12 weeks prior to randomization. Okay, so on the next slide, I'll go through some of the key endpoints that we will be studying in Forzetto. So first, we have our primary endpoint, which is a change from baseline in rise from floor velocity at week 73. And in the box below, you can see that we have a number of select secondary endpoints, including the key functional endpoints of stride velocity 95th centile, the North Star ambulatory assessment, the 10-meter walk-run velocity, and forced stair climb velocity. We also have a number of performance and patient-reported outcomes, including a functional composite score, which is essentially a combination of a number of other functional endpoints that are put into a single score so that we can have a global unified view of how zolbetuximab rostudirsen might be impacting function. Lung function is assessed via forced vital capacity, percent predicted. And we're also looking at the patient global impression of severity and the patient global impression of change. We're also looking at safety and tolerability and some other endpoints, including blood creatine kinase, some pharmacokinetic endpoints, and anti-drug antibodies. On the next few slides, I'll dive into a little bit more detail into specifically what we're looking at with these different endpoints. So for the selected clinical outcomes in Forzetto that measure function, we have our primary endpoint, rise from floor velocity, which essentially is the speed of rising from the floor to standing. So how fast a participant can rise from the floor. SV95C is the 5% fastest steps taken during everyday living. So a participant will wear a device that kind of looks like a smartwatch, which measures the speed of the steps that they take outside of the clinic. So at home, at school, on the playground, etc. We're also measuring NSAA, which is a 17-item scale measuring motor function. And we're looking at 10-meter walk-run velocity, which is the speed of walking or running 10 meters, and the forced stair climb velocity, which is the speed of walking up 4 steps. And finally, we're looking at forced vital capacity, percent predicted, which is a global assessment of lung function. With respect to patient-reported outcomes in Forzetto, a couple of the PROs that we'll be measuring include the patient global impression of severity, which is basically how a participant feels about the severity of his condition on a scale of 1 to 5. And the patient global impression of change, which is how a participant feels about any change in his condition from the start of the study to another point in time during the study on a scale of 1 to 7. And finally, some other selected outcomes in Forzetto include safety and tolerability. So the measuring of and this collection of treatment emergent adverse events, which is essentially all side effects that a participant might experience while receiving the drug or placebo. We'll be measuring blood creatine kinase, which is a marker of muscle injury, as well as pharmacokinetics, which are endpoints that measure what the body does to the drug. So, for example, what the concentration or the half-life is in the plasma. And we'll also be looking at anti-drug antibodies, which are antibodies that a participant might produce that bind to the drug. And so with that, I'll finish on this last slide here that cover some of the key recent milestones for zolbetuximab rostudirsen. And that include In Q1 of last year, we completed the enrollment of our Deliver registrational expansion cohort that was used to generate the data that we used for our BLA filing. In December of last year, we reported out on positive top-line results from Deliver. And in May of this year, we initiated our phase 3 Forzetto study, which we've just gone over today. In the same month, we also submitted our BLA to the US FDA, seeking accelerated approval. And looking ahead a little bit, in Q1 of next year, we have our potential US launch of zalesiment rostudirsen with a PDUFA date of January 21st, 2027. And if successful, would represent a first potential launch of Dyne using a medicine that incorporates our FORCE platform that was designed to directly target the tissues most impacted by Duchenne. And so with that, I will thank you for your time and attention and answer any questions you may have. Thank you very much.
Maria, thank you. That was quite a good walkthrough of the trial. I've got a couple to begin with, and I know that Liana's going to join me and ask some questions as well. The site is active at the RDR site in North Carolina. Do you have other US locations in mind that you can talk about?
We do have other locations in mind. They aren't activated yet, so we won't disclose at the moment what they are. But certainly, as soon as they are activated, we will make sure to post that on clinicaltrials.gov and certainly let you know. We are excited to have more than one site, of course, but we are still in those planning stages.
I've seen that. I checked clinicaltrials.gov this morning, but before the webinar, there's no mention of where this might be internationally. Have you any guidance to give out about where you're thinking about?
We are thinking of a number of countries outside of the United States, which include Europe and some other regions in the world.
Okay, thank you for that. Given the fact that you've got 72 weeks of placebo, is the company's expectation that it will be a vast majority of patients ex-US that will actually power this study?
Most likely, yes.
Mhm.
We understand that 72 weeks is a long time for a placebo-controlled period, but based on the data that we've seen from Deliver, we wanted to design a phase 3 study that could actually show benefit in function. And so that was the design that we landed on. But fully understanding it is a long time.
Yeah, very good. The CureDuchenne Cares team travels widely, not just in the US, but abroad. And I can tell you personally that we've seen an enormous unmet need in Mexico, in China, in Australia, in Asia, so I think there'll be real interest in all of those countries in order to participate in this study.
It's great to hear.
Yeah, my last one before I hand it over to Liana is I didn't see, maybe I missed it, but maybe you're not doing it. Is a biopsy part of the study?
There is no biopsy.
Okay.
And the reason is that we had biopsies in Deliver, and as you mentioned at the beginning, saw some really impressive results on the dystrophin there. And the focus of phase 3 is really to understand function.
Yeah.
So that was the reason why we're not doing a biopsy.
Yeah, for me, it was interesting that the 12-month dystrophin data clearly exceeded by far, what you'd seen at 6 months. And just trying to understand the pharmacokinetics and dynamics of this drug, is 4 weeks dosing interval optimal? Should it be 6 weeks? Should it be 8 weeks? So longer term biopsies could really lean into that for you guys.
Yeah, that's a great question. Certainly, it's something that we could potentially look at. We did look at 8-week interval in the Deliver
Mhm.
study, but based on those results, the 4-week interval was selected as the one for registration.
Okay, thank you for that. And thank you for the answers. Liana.
Yeah, thank you. I noticed some questions are coming in. I'm just gonna remind people that if they have a question to put it into the Q&A rather than the chat. We're monitoring the questions there. So just to get back to Maria, you did a great job of outlining the trial. I'll just go through some of the points that you made. So because we had some questions come in ahead of time. You said washout for eteplirsen is 1 week,
Yeah.
12 weeks for givinostat or other things. In terms of steroids, is it allowable to be on prednisone, deflazacort, or vamorolone? I was questioning about vamorolone.
Yes, vamorolone is allowed in the study.
Okay, so as long as that's a stable regimen.
That's right, either a daily or a weekend regimen. Um, the intermittent regimen is not allowed in our per protocol. So we do ask, and this is based on clinical data that's been published previously, suggesting potential more favorable outcomes with those two regimens. So we just want to make sure that we're sort of, you know, kind of evening the playing field for patients or participants who are in the study that they're on an optimal steroid regimen is determined by their physician.
Terrific. Thank you. We've had some other questions here about plans for non-ambulant individuals. So it's interesting, right? The phase 1/2 included ambulatory and non-ambulatory, correct?
That's correct.
But the phase 3 is going to be in ambulatory individuals.
This phase three, yes. Just because again, we need to move our attention to measuring an effect on function.
Mhm.
And so until I think we can identify an outcome that's uniform across both populations of participants, this is the study that we're focusing on now.
Excellent. The question is specifically asking if the drug is approved in January, could it cover non-ambulant individuals? And, I think the answer to that, and correct me others if you're wrong, is like we'll have to see, right? But there's data that's been collected in the phase 1/2 in those individuals. So we do know dystrophin levels in those individuals. And we'll have to see what the FDA decides in terms of the label.
Exactly.
Terrific. Mike, is there any you wanted to
Yeah,
jump
there's
in with?
one. Thank you for those, Leanna. There's one that caught my eye. The inclusion criteria in age is 4 through 18. And whenever I look at what's going on in gene therapy world today, we're taking young children from 1 to 2 years of age. Was there any consideration by the company to go younger than 4? Any particularly with diagnosis, now in newborn screening?
Yeah, that's a great question. We would love to be able to understand the effects of xeroacidosin in a younger patient population. So that's something for us to look at,
Yeah,
certainly.
we're asking the exact same question of gene therapy, so I feel it's only fair, with a therapy like this that we know puts in ostensibly a very, very large 90+% size of the protein, what that would actually do in a young person.
Yeah, we have the same questions and would love the same answers.
Okay, I'll keep it easy. I'll not ask any more hard questions.
There's another exclusion criteria question here asking about left ventricular ejection fraction and whether or not that is on the exclusions for the trial.
There is an exclusion around that, yes. Yeah. So there are more details, certainly. If there are people out there who are interested in the study, we do encourage you to speak to your physician and connect with an active site to learn more about the study.
Terrific.
Last ones, Liana? Any more?
Well, there's the question that every questions people always want to ask about developing programs for other exon skipping, other exons. And I know Dyne has talked about it publicly, so I feel like that might be a safe question to ask you, Maria.
Yeah, so it's a great question. We're really excited that we have four additional exon skipping programs that are in our pipeline. So that would be for people with Duchenne with genetic variants that are amenable to skipping of exon 53, 55, 44, and 45.
Mhm.
And so we are excited to move those through the pipeline.
Terrific. We've had a lot of more questions coming in just while we were talking. So it says here, you mentioned something in the opening about nearly full-length functional dystrophin. And so can you speak a little bit more to that? It's sort of just a very foundational question. Um,
Yeah, absolutely. So the concept of exon skipping is that we are we're removing something to make something whole, which is a little counterintuitive, right? So I think you have well, probably a lot of people on this call are really familiar with the different mutations or the genetic variants that cause the Duchenne muscular dystrophy. And so the idea is that if there are genetic variants that are deleting parts of the Duchenne gene, the DMD gene, then by removing exon 51, you can allow the restoration of that open reading frame. So it's you're keeping most of the length of the protein itself. There will be parts of it that are missing either because of the genetic variant or because of the exon skipping that Z-rostudirsen enables. But the idea is that it's a relatively small portion compared to the entire length. So it isn't full length, but it's probably as close to full length as you might be able to get under these circumstances.
And I'll add a little bit in, Maria. This ostensibly, depend upon the mutation, is a protein that we actually see expressed in nature in Becker muscular dystrophy patients. And for the most part, that there is either much milder or asymptomatic type. So we're reducing a protein that we know is functional, depend upon the mutation that we're actually skipping with exon 51.
Thanks. Any others, Mike, that you thought you flagged that have come in?
No, I'm good. I think that this is, I think what people are looking for now is just the recruitment to start and seeing this across other countries, because as I said, Maria, there is an enormous unmet need once you walk out of the US, and that we've talked about some of the countries we personally have visited. So I look forward to seeing this here trial really get into gear.
Thank you.
Is that it, Leanna? I'll wrap up if there's no more questions.
Sure.
Okay, so thank you all for joining the CureDuchenne webinar today, and a big thank you to Maria and the whole team at Dyne Therapeutics for the work they've done developing this drug and for sharing the important update with us. This is an exciting time for the Duchenne community and the company. Z-rostudirsen has the potential to treat up to 13% of the Duchenne population, and we've seen statistically significant and robust increases in dystrophin expression and a favorable safety and tolerability profile. We look forward to the FDA review and the potential new product launch early in 2027. Post-webinar email will be sent out to all attendees tomorrow, and we'll let you know when the webinar is posted online for viewing and sharing. And just thank you again to Dyne. Thank you all for joining us, and have a wonderful rest of your day.